lactic acid as their ferment, part of their ferment. All right, fermented foods, right? Be really amazed. There's a great, well, some of you watched it. I already gave it to you as extra credit, right? The cooked episode with the fermented food. If you didn't watch that, you can still go back and watch it, just for fun. But if you like to eat, you'd be really amazed at how many foods are fermented. I mean, who knew chocolate was fermented, right?
What?
I know, see, I know who didn't watch the video, but people. So sauerkraut, pickles, yogurt. You can pickle food by simply adding acid to them. But you can also add the bacteria, let the bacteria produce the acid, and that will slow bacterial growth as the acid goes up and preserve the food. We also know that if you ferment some things, you can spoil food, right? So wine, the whole point of pasteurization was to prevent the fermentation of lactic acid or acetic acid in wine. So fermented food can preserve food, but also in some cases it can spoil food. You want to have the right bacteria to do the job that you're trying to do. In the case of beer and wine, you want yeast to make alcohol. You don't want bacteria making acid. Question?
Good question. So a lot of times you see that wine will contain sulfites. Is that
To prevent bacteria from growing.
okay? Yeah.
A lot of people say it irritates them or gives them headaches, right? People don't want the sulfate. Okay, alcohol fermentation. In the case of alcohol fermentation, pyruvate is going to lose the carbon dioxide. I think I have a picture. Easier with a picture. All right, so here's glycolysis. Each one of these pictures is organized a little differently, but if you can tell yourself the story from each picture, then you've really got it down. So here's glucose going through glycolysis to form pyruvate. We've made 2 NADHs, but now we need to regenerate these. So what we're going to do is we're going to lose the CO2. We're going to make acetaldehyde, and then this molecule, you can see it's getting reduced. See how it's gaining hydrogens? And so when this acetaldehyde gets reduced to ethanol, NADH will be oxidized back to NAD+, and now it can participate in multiple rounds of glycolysis. Now, both ethanol and CO2 are actually important, right? So CO2 gets produced in alcohol fermentation. It's what causes bread to rise, and champagne to be bubbly, and beer to have carbon dioxide in it, right? So the carbonation of alcoholic beverages usually comes from this carbon dioxide being produced in fermentation. And then, of course, the alcohol is also a byproduct. You don't get one without the other, right? If you make CO2, you also make alcohol. If you make alcohol, you're also making CO2 because the two are both byproducts of the same chemical reaction. All right. Well, it's been a hard day to create.
Other than the smell of your feet, which we've covered already, this is probably the most popular cheese-related question on the internet. And the answer to why Swiss cheese has holes is actually pretty simple. All cheese is the product of fermentation, a type of respiration that some microorganisms use to feed in the absence of oxygen. Their food, in this case, is lactose, the sugar found in milk. And to make cheese, all you really need to do is warm up some milk, toss in some bacteria, wait for it to thicken into chunky curds, and then give the whole thing a brine bath to make it form a protective rind. Then you just let the bacteria do their thing somewhere safe, like a cheese cave, and then a few months or years later, boom, you've got sandwiches. Now the holes have to do with what kind of bacteria you have in your cheese. Some cheeses are made with homofermentative bacteria. They tend to produce one main byproduct when they respire, in this case lactic acid. And in addition to certain cheeses like cheddar, they're used to make pickles, sauerkraut, yogurt, and other tart-tasting things. But there are also heterofermentative bacteria, which create a whole bunch of byproducts, including carbon dioxide. These bacteria, like P. shermani, are what are used to make Swiss cheese. So as the bacteria are in their little cave or whatever, eating lactose and excreting carbon dioxide, the gas gets trapped inside the cheese rind and forms bubbles. If you're making a soft cheese, these gas bubbles can move through the cheese and leak out of the rind. But Swiss cheese is more dense with a thick rind and the gas can't escape, so these bubbles form air pockets which eventually leave behind the cheese's signature holes. Cheesemakers refer to these holes as eyes, and they can control the size of them by adjusting the cheese's acidity, temperature, and aging time, all of which influence the byproducts of the bacteria. So for example, milder baby Swiss has smaller holes because it doesn't age as long. And here's a fun fact. In the US, the Food and Drug Administration actually regulates the size of Swiss cheese holes. Holes must be between 3/8 of an inch and 13/16 of an inch in diameter so they don't gum up the commercial cheese slicers. Who knew? Thanks for asking, and thanks especially to our Seppable subscribers who keep these answers coming. If you have a quick question, let us know on Facebook and Twitter.
Maybe they can get the Trump administration to work on deregulating Swiss cheese holes. And get them busy with that, and they can leave the EPA alone.
Oh,
All right, sorry. I really try hard not to make these editorial comments. It's very difficult. All right, so that's kind of a cute video, right? Swiss cheese, fermentation is all related. So typically when we think about alcohol and CO2, it's often done by yeast actually, which are a eukaryotic microorganism. So with sugar and yeast, usually gives us wine and beer. And so here's a mixture of sugar and yeast. And you can see it's bubbling because they're producing CO2 gas and foaming. And this combination is actually used to make bread as well as beer, big kegs of fermentation happening. So long before we understood this process, we've been using it, right? I mean, we've been making sourdough bread since ancient times. The ancient Greeks, the Egyptians, fermentation's been happening for years, and alcoholic beverages too. How many of you have ever made sourdough bread from sourdough starter? Yeah, a few of you have. I used to do it for a long time, and then I got tired of the sourdough starter like having a pet. You have to take care of it all the time. But this is a kind of a fun video too.
When we think of sourdough bread, lots of us think of San Francisco or maybe Alaska, but the history of these tiny loaves goes back further than the California gold rush. We think ancient Egyptians were eating sourdough too, making it the oldest leavened bread, which is bread that has something in it to make the dough rise. For sourdough, that something is a mix of wild yeast and bacteria, which break down sugars and make chemicals that give the bread its chewy, sour taste. Generally speaking, bakers make all bread dough rise using yeast, but the source of the yeast is what makes sourdough special. Nowadays, you can buy little foil packages with Saccharomyces cerevisiae yeast, a species that we use in baking and brewing. But before grocery stores, people had to culture their own wild yeasts, keeping them alive in something called a starter. You can make sourdough starter by mixing some flour and water and letting it sit out, because the air around us is full of microbes like yeast and bacteria. After some time, different species will make their home in the goop and keep multiplying as long as you keep feeding them. Microorganisms like yeast and bacteria break down organic compounds in a process called fermentation, producing other chemicals like gases or alcohol. So when yeasts digest the sugars in flour, they make bubbles of carbon dioxide gas, which makes bread dough rise. The bacteria that end up in the mix are usually part of the Lactobacillus genus, which includes species that we use to make yogurt and cheese. These bacteria break down sugars into chemicals like lactic acid and acetic acid, giving sourdough its signature tartness, and different species produce slightly different flavors. For example, in 1971, microbiologists Leo Klein and T. Frank Sugihara were researching San Francisco sourdough starter, and they found that the most common yeast species were Candida milleri and Saccharomyces exiguus, while the main bacterium was the never-before-seen Lactobacillus sanfranciscensis. These yeasts and bacterium have a special relationship. Unlike the yeast S. cerevisiae, these yeasts are really good at surviving in the acid that the bacterium produces, and they don't digest the sugar maltose. But L. sanfranciscensis needs maltose to stay alive, so these microbes can live and grow without fighting over resources. As long as you keep adding flour and water, a starter can live for years, and different flours, temperatures, humidity, and feeding schedules can change how your bread turns out. So next time you're biting into a delicious sourdough loaf, be sure to thank the microbes that make those flavors possible. And thanks to all of our patrons on Patreon who make...
Okay, so it's a great story, and if you've made sourdough bread, you probably know the story a little bit already. So there's two microorganisms both fermenting the sourdough bread. You have a lactic acid producing bacteria, a lactobacillus, that's named, if it's the San Francisco sourdough, San Franciscensis, I have to say that slowly, and this is making lactic acid. The yeast, the Saccharomyces exiguus is making carbon dioxide and alcohol. So you have three different fermentation products in your sourdough bread. So the lactic acid gives it that sour taste, and the CO2 causes it to rise, and the alcohol gets burned off in the cooking. Are you with me so far? So if you want to make your sourdough bread more tangy, then you want the bread to rise slowly to give the lactobacillus more time to produce lactic acid. If you let the bread rise in the refrigerator, you favor the bacteria over the yeast. If you want your bread to be less tangy, you can let it rise at room temperature or even in the oven, and then the lactobacillus won't have a chance to make a lot of the acid, but you'll get the bread to rise more quickly because you favor the yeast. So you can actually kind of control the flavor of your sourdough bread by the temperature you let it rise at. So people who want really ultra tangy sourdough bread, so really great bakers, they do it in a cool room and they let it rise maybe over a couple days in the fridge, and they get a much tangier sourdough bread. So what are they doing? They're not changing the ingredients at all, right? They're simply changing the growth traits. They're adapting their incubation. I mean, as a microbiologist, I think of it as they're incubating their bread at a different temperature to favor one organism over another organism. It's kind of a cool story. Because this is a sourdough starter, and because you produce CO2, lactic acid, and alcohol, the alcohol rises to the top. So you see this little layer here? People who make their own sourdough bread call that the hooch, because it's full of alcohol. Okay. Sourdough bread's a lot of fun to make, but you have to have lots of free time. You can actually take the starter and you can make pancakes out of it, muffins. I mean, if you've got time to do it, I found it very hard to have 2 cats, 2 children, a husband, and sourdough bread. It was like a whole nother person that needed attention. Okay. One of the things I said at the beginning is that we were focusing on 2 kinds of fermentation, lactic acid fermentation and alcohol fermentation. But I want you to know that bacteria can produce lots of different products. So depending on the organism, lactic acid and ethanol are common, but like the Propionibacterium, these are the ones that cause acne. They tend to be associated with acne. They can produce propionic acid and acetic acid. Clostridium, which smells awful, right? We use Clostridium in the lab. It produces something called butyric acid, and that's one of the reasons it smells so bad, because butyric acid is just awful, noxious smelling. E. coli and salmonella can produce succinic acid, so that's pretty smelly. They can also produce hydrogen gas in the process. Enterobacter can produce butanediol and acetylene, and actually we're going to be looking for some of these molecules when we do the ID project. When you have to tell the difference between whether you have Enterobacter or salmonella, sometimes finding out what the fermentation products are is a way to figure out which bacteria you have. So we're going to take advantage. I'm going to give you the opportunity to test for some of these molecules to figure out who your unknown is when we get later on in lab. All right, so fermentation is pretty awesome. All right, regardless of what byproducts are produced, the purpose of fermentation is all right, last chance to click in. All right, so usually I have A and D, but D is really the correct answer for the test, right? Even if you believe very passionately that purpose is one of these food-related items, on the exam you want to select to produce an AB+, because that's the cellular reason for undergoing fermentation. All right. Before we wrap up this topic, remember that I said that glycolysis is a central pathway? So metabolism in cells is really complex. So I'm going to show you this really quick and then we'll go back. This is a cell where biochemists mapped a bunch of the pathways. So here's glucose going to form, this is the Krebs cycle, right? These are all the byproducts. But what I want you to point out is that there's a lot of other pathways in the cell besides this one that we just studied. So why the heck make you learn one of them when it seems like there's no reason, right? There's so many, it's impossible to learn them all. And it is hard to learn them all. But the reality is that if you learn about glycolysis, you actually know the most fundamental pathway in the cell. Because almost all the other important things that the cell does come off of glycolysis in the Krebs cycle. So for example, here's let's start here. So here's our carbohydrates and glucose, here's glycolysis. If you're going to make proteins, if you're going to break down proteins, amino acids can get broken down and they get shuttled into the different parts of this pathway. If you're going to break down fat, glycerol gets converted to glyceraldehyde 3-phosphate, and fatty acids get sliced into 2 carbon pieces and go right into the Krebs cycle. So even though we have all these other molecules, they can get broken down and then use the same pathway you just learned. It's efficient, right? Not even if you don't agree. And then by the same token, I don't know if this does a good job, but it goes the other way. So you can make proteins, these pathways all go the other direction. So if I want to make proteins, I can make many of the amino acids by taking intermediates from Krebs cycle and from glycolysis, and they can be converted into protein. So if you're short of a protein or short of amino acid, we can take this pathway and we convert these molecules the other direction. We can take acetyl-CoA and glycerol-3 phosphate that are produced here, and we can turn those into triglycerides and store that energy for later. Right? Evolutionarily, you had periods of feast and famine, so being able to convert energy that was in excess into triglycerides for storage was really important for survival of species. So while this is just one pathway in and amongst many in the cell, it's the pathway. And all the other important things the cell does really branch off of glycolysis. And I'm not showing you all of them, but you can make the basics of nucleotides come off of these pathways, all of the amino acids that we can make, all of these molecules can be built from the intermediates in glycolysis in the Krebs cycle. So it's a major pathway. It is the one to know. Okay? That's a cool picture, right?
Sure.
You guys are like, turn it off, turn it off. All right. In the lab, it's actually the new big incubator is kind of in front of it. There's a whole bunch of these like big pathway charts where biochemists have charted all this stuff. Okay, the study guide says, 2 more questions here. Describe 2 main types of fermentation. What are the 2 main types? Lactic acid fermentation and alcohol and CO2, right? What are the starting materials in both cases? Pyruvic acid. It's the same starting material. What are the products in both cases? The product of lactic acid fermentation is?
Lactic acid.
The product of ethanol fermentation is?
CO2.
Ethanol and CO2. Okay, so you make sure you get both. Do you see that there was a lot of questions in these 2 sentences? To get full credit on an exam, you really have to address all of it, right? Both kinds, both sets of products, and the fact that pyruvate is the same starting material. All right, now I talked about this a bit ago. If you're baking sourdough bread and you want a more tangy taste, what might you do?
Refrigerator.
You're going to let it rise in the refrigerator because you want to favor what? The lactobacillus can grow better in the refrigerator than the yeast, and so it's going to allow more lactic acid to be produced. I think it's cool that something as yummy as sourdough bread that you can manipulate the taste by how you treat the microorganisms in the food. I think that's really neat. Okay. I know you're happy that we are done with metabolism. But if you're struggling with the material, you want to make sure that you spend some time on it now, because on Thursday we're going to dive into genetics, all right? And talk about DNA and RNA and protein and how basically the information in your DNA, how it's used to build cells. And that is a big topic too. And both of those are on the next exam. They're 2 fairly weighty topics. And so you don't want to lose what you've learned in this part. You want to make sure you are solidifying that understanding even as we move on to the next topic. All right, should we talk about cholera for a moment? So cholera, I have a subtitle, The Death by Dehydration, because when people die of cholera, that's what happens. They die of dehydration. And there's a great video, which I'm not going to show you because it's kind of long and you can watch it on your own, but it kind of goes through what happens in a community when people get cholera. But cholera is caused by a bacterium, so it's not a virus, it's a bacterial infection. It's gram-negative. It's a comma shape, it's a vibrio, so it's got this nice electron micrograph. It's got a nice flagellum. And what cholera does is it produces a toxin that when it gets into your intestines, it messes up the electrolyte balance and basically causes your intestines to flood with water. And that results in massive amounts of diarrhea. Vibrio cholerae, the bacteria actually is a symbiont with these marine copepods. And so Vibrio cholerae didn't start off as a pathogen of humans, but it spilled over into humans. And so it seems to have originated probably near Bangladesh in very kind of brackish water that was there. And while people weren't hanging out in that water, it wasn't an issue. But as people, continue to clear the land and kind of drain the swamps and build more land, you have people coming in contact with the marine water, the brackish water, but also these copepods. And what happened is probably the first million times people ingested Vibrio, nothing happened. But then at one point, one of these Vibrio was making a toxin that caused the massive diarrhea. And it's a very efficient way to spread yourself around. Because once someone sprayed diarrhea everywhere, it contaminates the water. Now lots of people ingest that bacteria and get lots of chance to divide and reproduce. It becomes an evolutionary advantage. And so because of the toxin producing all this diarrhea, there are several species now, two in particular, Vibrio, that really cause pretty horrific disease. So this spillover happened, and then by the 19th century, it became pretty widespread. As I was mentioning, this toxin made it really easy to adapt to humans. And the first pandemics occurred in Calcutta around, I want to say 1817 was about the first ones. And then at that point, by that time, we were traveling by sea everywhere, right, all over the world. And so Vibrio just hopped on, just like the plague bacterium hopped on ships, the cholera bacterium also hopped on ships. And then if you're interested, there's actually a great book called Pandemic by Sonia Shaw. She talks a lot about the history of cholera, but she talks about cholera arriving in New York City and the boroughs in New York City and the poverty and where cholera hit the hardest. But the fact is that people would arrive on these ships and they would have to put a yellow flag up if someone had cholera. They wouldn't let anybody disembark if they had cholera on the ship. But eventually there were no sewer systems, and so eventually cholera made its way into New York City. And it hit, as you might have expected, the poorer parts of the city first, where people were just on top of each other in filth, because New York used to be pretty horrific at this period of time. But cholera is an equal opportunity killer. And because sanitation wasn't employed everywhere, or anywhere at this point, eventually the cholera bacteria got into all of the neighborhoods. And so anybody in New York could die of cholera. And it really didn't get cleaned up until we cleaned up the water, because it's a waterborne pathogen. Remember the Broad Street pump and Jon Snow and the Thames? So I also use words like filth and dirty water and poo at one end of the water, going downstream, being the drinking water for other people. And so that's Vibrio heaven. That's how it can divide and reproduce and get to more people. There's an interesting story in Pandemic about New York City, and some of the politicians and the public works people were trying to get the water, rather than get this brackish water from where the ships were coming in that was always contaminated, they were trying to get water from an area outside of New York, and it was going to flow through gravel, which actually makes for a fairly nice filter, and it would have been much cleaner water. And there was a bunch of people, you'll recognize this, people were making money off of selling water locally, and so they blocked the project, and so people kept dying of cholera. There's things you can count on, and politics, as usual, is one of them, I think. All right, so cholera. How does cholera transmission happen? Just drinking contaminated water. And again, it causes such severe diarrhea that people end up, it causes vomiting too, but it's famous for diarrhea. You end up just having diarrhea everywhere and it gets into the water. You can get it from food that's been mixed with water that's also contaminated. Imagine, for example, some of the places in the world where the formula companies that make powdered baby formula, they want them to mix the baby formula with water and feed it to babies. And so, and of course, if the water is contaminated, you can kill the babies that way, right? There's a lot of public health stories that kind of go in and among how do you prevent people from selling their formula. The United States isn't a good market because most women breastfeed now, or many women breastfeed. But anyway, contaminated water is a big problem because you can mix it with food. We still see cholera where there's inadequate sewage and water treatment. So the last couple of big outbreaks have been in Haiti after the 2009 earthquake. That earthquake resulted in a whole bunch of people coming in to help rebuild Haiti and provide medical care. And in the process, some of the aid workers brought cholera with them, not intentionally, but they weren't using good sanitation. There isn't good sanitation infrastructure built in Haiti, and so a whole bunch of people then died from cholera a couple of years after the earthquake. And then the current outbreak, the biggest current outbreak that I know is in Yemen. And I haven't looked at the current numbers, but that's been happening over the last couple of years because there's a war in Yemen. And then you can't get in there to treat people because of the war. Okay, I was talking about New York City, and I sort of told you this story a little bit. The first was in 1817, I said, in Calcutta, and then there's been 7 documented pandemics of cholera since. The story of New York, it's too long to talk about, but it's pretty amazing, the spread of cholera in New York City and how people responded. Cholera, you can be healthy in the morning and really deathly ill by the evening. You can be dead by the evening, honestly. It causes vomiting and severe diarrhea. The dehydration is so severe that one of the signs is tenting of the skin. So when you pull up the skin doesn't just bounce back, it stays up. Sometimes people check for hydration in animals that way. It's a little easier, they have soft, looser skin, but you can see it in humans too. It can kill you within hours. The rapid weight loss is from the water, not from losing fat, but it's just from losing so much fluid so quickly. And it's pretty gruesome. So on a typical year, there's 3 to 4 million cases, but there's many that are unreported. Some people get cholera and they don't get that sick. There's a vaccine for cholera, and they're trying to vaccinate vulnerable populations. You have to get 3 doses to get fully protected. It's about 65%. But you can get some immunity is the take-home message. And so some people don't get that sick from cholera, don't report it, and then of course some people die from it.
If it's a bacteria, how is there a vaccine for it? How doesn't it work?
There's vaccines for lots of bacteria. They can take, you can kill the bacteria and put it in a vaccine, and then your body will still recognize the outside molecules. Sometimes they make subunit vaccines where it's just little pieces. The cholera vaccine is an oral vaccine, so it's very easy to give. Yeah. 100,000 people die a year on an average year. You know, cholera is closely associated with social development. So if you have treated water, chlorinated water, if you have sewage systems, cholera just isn't an issue. But where you don't have those things, cholera can be present in the water. So this is why Yemen is such a big problem right now, because not only is there a civil war going on in Yemen, but you can't get healthcare workers there. So you can't even treat the people who are getting sick, because there's actually good treatments for cholera. If you give people oral rehydration therapy, they shouldn't die from cholera. Cholera is easy to treat, but you have to be able to get there to treat people. I updated this morning. This is from the World Health Organization. The most recent data is from 2016. They don't have a lot of the Yemen data here yet, unfortunately. That was what I was hoping to see. This is the outbreak in Haiti. The earthquake was in 2009, right? And the aid workers came in, and then you see this big outbreak. The purple is the Americas. So lots of cholera in the Americas at this point. And there'll be another spike in a couple of years when they have the Yemen results included in there. All right, so you can detect cholera from a stool sample on media. So this is on thiosulfate, citrate, biosalt, sucrose, and it ferments the sucrose, and so it turns bright yellow. And so this is selective for Vibrio organisms, actually, and then the yellow color shows the fermentation. So this is the main thing about cholera that I don't think people realize is we're super easy to treat. So if you catch somebody early, all you need is a little pack of these oral rehydration salts. You put them in clean water. It replaces the fluids and the electrolytes. And that's all that you really need to give them. So that works for most people who are really sick with cholera. If you replace fluids, fewer than 1% of people will die if you get fluid replacement. You can also give someone antibiotics, and if someone is really severely ill, they'll give them antibiotics and they'll give them fluids by IV. So someone who's more severely ill, that they can't just give them the oral rehydration therapy, they can actually treat them with IV fluids. You see the cot that the different people are laying on? See the hole in the middle? So the diarrhea is so severe that what they do is they put a bucket under the hole. So that as you're laying in bed too weak to move, the diarrhea goes into the bucket. It's liters and liters and liters. They call it rice water stools because you lose so much fluid with the diarrhea. It's pretty horrific. But again, it's actually fairly easy to treat, and there's a vaccine. And so we're trying really hard. If you look at the World Health Organization, they have a plan to get rid of cholera as a scourge. I think it's by 2020. I don't think they'll get there. But the goal is to get all the people in vulnerable areas vaccinated. And then if you get it seems about 60%, if you're fully vaccinated, about a 60% reduction in your chance of getting cholera. Just by reducing the number of cases, you get some herd immunity, and then it'll just reduce the number of people dying by a ton. All right, prevention. So I talked about the vaccine. What about here in the US? So Vibrio bacteria are kind of famous in general for contaminating shellfish. So these organisms, the shellfish are filter feeders, right? So they filter the water. And if there's bacteria like Vibrio in the water, they can actually pick up the toxins, and they can actually pick up the bacteria. So shellfish is actually really carefully monitored in the US. If you serve shellfish, I know this because I'm teaching the food safety. If you serve shellfish in a restaurant, you have to save the container until all the shellfish are gone, and you have to save the tag for 90 days after people have eaten, which if that's like a regular dish, you have a lot of tags that you have to keep track of. Because enough people get sick from eating shellfish that they have to keep track of all of this. But the U.S. tests all of the imported and domestic shellfish for cholera. When there's cholera outbreaks in other places in the world, USAID first provides medical supplies and those oral rehydration solutions, and now they're starting to provide the vaccine. So the idea here is that we're going to try to keep people from dying where we can of cholera because it's possible. But if you can't get there because the political situation is too unstable, it's too dangerous for aid workers to get there, then people will still die. And in Yemen, that's kind of what's been happening. All right, so our modern water treatment situation has saved us from having to deal with this now in the U.S. I love this. It's an 1865 poster. So this is in the middle of all of these cholera outbreaks. And it's just everything on here is wrong. Notice, preventatives of cholera. This is the Public Health Department. Published by order of the Sanitary Committee. Be temperate in eating and drinking. Avoid raw vegetables and unripe fruit. Abstain from cold water when heated, and above all from ardent spirits that don't drink. If habit have rendered them indispensable, take much less than usual. If you can't stop drinking, try to reduce it. Sleep in clothes warm. Do not sleep or sit in a draft of air. Avoid getting wet. Telling people who have infectious diseases not to bathe. It's just bad advice. Attend immediately to all disorders of the bowels. Take no medicine without advice. Anyway, I'm sure that we give patients a lot of instructions now that we're going to have to laugh at. 100 years from now, people will laugh at what we tell patients, right? But it's kind of fun to make fun of their old posters that have nothing to do with what we know about how cholera is transmitted. I think that's my last